So I'm going to start by diving into the fascinating world of recombinant DNA technology, which is actually the cornerstone for modern biotechnology. Recombinant DNA is essentially a hybrid DNA molecule, and it's created artificially by combining DNA from one or two or more different sources. This technology allows scientists to produce large quantities of specific genes or proteins, which can then be used in various biotechnological applications, including the development of pharmaceuticals and genetically modified organisms. But to understand how recombinant DNA is made, we're going to go through the steps involved in making it. This can be a bit of a difficult process to remember, so try to focus as I go through it. It's okay if it takes a couple of times to understand fully. But it will also be really helpful for you to have a good understanding of the process of DNA replication from our previous unit if you're struggling to grasp this content. So if you are struggling, I would absolutely recommend reviewing some of the previous stuff. I'm just going to get a drink and then I'll go through the steps. Okay, so let's walk ourselves through this diagram. The first step in creating recombinant DNA is the isolation of the target gene. This is achieved using something known as a restriction enzyme, which is a specialized protein that cut DNA at specific sequences known as recognition sites. They're basically made, you'll learn about restriction enzymes and their purpose in higher level, but just know that they essentially cut DNA. They're able to do that. But they do not cut the DNA through the hydrogen bonds as you would be used to with helicase if you know DNA replication. They cut it through the covalent bonds of the backbone. And these enzymes can cut DNA in two ways. They can do straight down, so resulting in blunt ends, or in a staggered manner, which produces sticky ends. Sticky ends are more desirable for biotechnology applications because they have overhanging sequences that easily pair with complementary sequences on the plasmid, facilitating a more efficient and stable integration of the target gene into the plasmid. When you think about DNA replication, you'll know that having an exposed nucleotide allows another base pair to come and connect to it using hydrogen bonding. And that's essentially what's happening here. It'd be really difficult if you have a blunt end because there's nothing to connect to it, and a covalent bond has to be formed. Okay. So in the next step, we need to prepare the plasmids, which are small circular DNA molecules found in bacteria. Unlike the chromosomal DNA of eukaryotes, plasmids are not essential for survival, but they carry beneficial genes such as those for antibody resistance. So you can think the reason they have these plasmids actually is to transport DNA between other bacterial cells. And this is essentially how bacteria develops immunity to certain diseases that are around it. So these plasmids are used as vectors to transport the target DNA into bacterial cells. The plasmid is cut with the same restriction enzyme that's used to isolate the target gene, which ensures that the end of the plasmid and the target gene are compatible for binding. This creates something that we call a plasmid vector, which is basically a vehicle for introducing new genetic material into a bacterium. The isolated target DNA and the prepared plasmid are often then combined in a solution with DNA ligase, which you'll remember from your previous studies is an enzyme that acts as a molecular glue. It's basically able to facilitate the formation of covalent bonds between the sticky ends of the target DNA and the plasmid. And this results in a recombinant plasmid. This recombinant plasmid now contains the desired gene and is ready for the next step. In the next step, the recombinant plasmid is introduced into bacterial cells through a process called transformation. The bacteria are treated with a solution that makes their cell membranes more permeable, often involving a brief heat shock, and this encourages the bacterium cells to take up foreign DNA, such as our recombinant plasmids that are in the same solution. Now, in order to, not all the plasmid DNA will be taken up, and there's no way that we can actually ensure that it will be. So in order to easily identify which bacteria have successfully taken up the recombinant DNA, the plasmid is engineered to include an antibiotic resistance gene. The bacteria are then grown on agar plates which contain this antibiotic. So only those that have incorporated the plasmid will survive. Basically, it's just ensuring that the plasmid has been taken up. If none of them survive, that means that the plasmid is not there because they are not immune to the antibiotic. In addition, we can also use fluorescent markers, which make the bacteria glow under UV light, and that provides visual confirmation of a successful transformation. It's important to note, however, that only 1% of the bacteria will typically take up the plasmid, and not all of these necessarily express the recombinant DNA correctly. And that's why we must use the agar plate. Okay, and the final step can only occur once the bacterial colonies containing the recombinant DNA are identified and have grown. The bacterial colonies are cultured to produce the target protein. So the bacteria transcribe the transferred gene into messenger mRNA or mRNA, which is then translated into a functional protein. After sufficient growth, the bacteria are lysed or split open to release the protein. The protein is then purified through various biochemical techniques such as centrifugation, filtration, and chromatography, ensuring the final product is suitable for intended use. This intricate process of creating recombinant DNA has revolutionized biotechnology because it enables the production of essential proteins and the development of new medical treatments as a result. Now, originally, the reason this was so profound is originally the small size of proteins essentially made it difficult to mass produce them. You could not do it through normal means. But recombinant DNA offered a solution through harnessing the body's pre-existing cellular process. It's a really interesting idea to use what the body has cultivated over millions of years of evolution to our own game. With that, that's a similar diagram showing the same idea. I actually think it might be a better representation of the final step. Sorry, I realized I didn't have that up. But I'll give you a second to take a look at that. Once again, you can see the stages that are happening. We isolate a gene from the human growth that we want. In this case, we've isolated a human growth hormone, and we cut that out using restriction enzymes. We then get a plasmid and we cut it using the exact same restriction enzymes, and that means the sticky ends are corresponding. That means that when we use ligase, where we add ligase into the same mixture, this gene and the plasmid are able to connect. Then using bacterial transformation, we get this recombinant plasmid to be intaken by the bacterial cell. And then bacterial cells have this really interesting quality where they're able to multiply rapidly. So they do that, and that results in a surplus of the protein which is contained within the bacterial cell. We can then isolate that protein for whatever applications we could use it for. Okay. So we're now going to, this is just another idea. Sorry, I didn't realize there were so many images for this. This is just showing the second process of that part. Once we have isolated the bacteria, this is the process, it multiplying. By including a antibody resistant gene or an antibiotic resistance gene and putting that antibiotic into the plate, we know that the only bacteria that are able to grow are the ones that have the resistance gene because they have uptaken the plasmid. So they multiply and they grow. We get a plate filled with these bacteria that we know have the plasmid that we need. And then we can take off that plasmid from the agar plate and isolate the protein.